All right, so I want to welcome everyone back. This is going to be our very first screencast on Chapter 14. And what we're going to do today is we are going to begin our discussion on the area of genetics. And before we begin that discussion, what we need to do is we need to talk about a very important man. And this man's name is Gregor Mendel. So when you talk about Gregor Mendel, what you need to think about here is you need to think about his use of the scientific approach to actually identify two very important laws of inheritance. So when we talk about inheritance, we're talking about the passing of traits from one generation to another. So Mendel discovered the basic principles of heredity by breeding a very unique plant, and this plant is the garden pea. And if you remember back to Honors Bio, we had talked about Gregor Mendel and we had talked about some of his experiments with garden peas. Now, some of the advantages of actually doing experiments with garden peas was that they have some very distinct heritable features. In other words, features that can be passed on to the next generation. And we call these features characters. And a good example of that would be the flower color that you would see in some species of garden peas. Now, we need to think about these characters as actually having variance. And then when we talk about variance, we're talking about differences between the character itself. And a good example, again, if we talk about flower color, is that sometimes you can find purple flowers in garden peas, and other times you can find white flowers in garden peas. And so overall, when you talk about these variants, sometimes you will see scientists use the word traits, and oftentimes you'll see that used in the mainstream as well. And another good advantage of using garden peas is that when Gregor Mendel was studying his pea plants, he had found out that the mating of these plants were very easy to control. So he was able to determine which plants he wanted to use in his breeding experiments. Each pea plant had a sperm-producing organ, or a stamen, and it also actually had an egg-producing organ as well, which was called the carpel. So he had the ability to actually manipulate each of these reproductive structures on these plants. And another thing that he had noticed about these garden peas is that cross-pollination could actually be achieved by dusting one plant with the pollen from another. So again, that allowed him a lot of control when it came down to his breeding of these garden peas and his study of genetics. So Mendel, what he did is he chose to track only those characters that varied in an either or manner. In other words, it was either this or it was that. He also used varieties that were considered true breeding. And when we say true breeding, it basically implies that these plants will produce offspring of the same variety as they would if the plant actually self-pollinated. So remember, these garden peas actually had both male and female parts. And the male part that would produce the sperm could actually fertilize the eggs of the same plant. And so if they self-pollinated, those offspring were actually clones of the parents. And so he only used true breeding plants in his experiments when he decided to actually cross different varieties of plants. In a typical experiment, Mendel would make two very contrasting or true breeding varieties. And he used the word hybridization when he mated these two contrasting varieties. A hybrid basically implies that you're taking the characteristics of one and the characteristics of a second one and actually putting them together, and sometimes you would come up with a new characteristic. The true breeding parents were identified as the P generation, and these were considered the parental generation, and that's where you get the P in this particular statement. The hybrid offspring of the P generation are going to be called the F1 or first filial generation. And when you take this first generation and actually have them self-pollinate, then what we would end up with is something called the first, or actually the second filial generation, or the F2 generation of offspring being produced. So in other words, you would start off with your parents. Those parents would produce, again, the F1, so that's going to be the F1 offspring or the F1 generation, and then those would self-pollinate, and we would end up with what we call the F2 generation. So those would be the offspring produced from the self-pollination of these F1 individuals. So one thing that Mendel had discovered through his many experiments with garden peas was something called the law of segregation. Now, if you just look at this statement, the word segregation or segregate basically means to separate. So when Mendel crossed contrasting true breeding white and purple flowered pea plants, all of the F1 hybrids, in other words, remember a hybrid is a cross between those two plants, ended up being purple. And you can see that over here on the right. So this would be considered his P generation. And if you notice, the P generation are considered true breeding. So in other words, if they self-pollinated, they would produce clones of the parents. So purple flowers were going to be crossed with white flowers. And so these are two separate plants. And when they were crossed, all of the offspring produced, in other words, the F1 generation, all produced purple plants. Now, when Mendel crossed the F1 hybrids, many of the F2 plants also had purple flowers. But what he noticed, which was kind of unusual to him, was that there were also a few of those plants that actually had reverted back to the white flowered plants that he had noticed, or actually he had used in the parental generation. So when you take these F1 generation hybrids, if you have them self-pollinate, then what you're going to find out is when the F2 generation is produced, and if you notice there are a few numbers down here, 705 and 224, and those become significant as we kind of continue our discussion, you'll notice that the majority did produce purple flowered plants, but there was maybe about a fourth that actually produced white flowered plants. And so that characteristic that was in the parental generation appeared again in the F2 generation. So Mendel discovered a ratio of about 3 to 1 purple to white flowers in that F2 generation. So Mendel reasoned that only the purple flower factor, in other words, that characteristic, was actually affecting flower color in the F1 hybrids. Mendel called the purple flower color a dominant trait. In other words, it overshadowed the white flower color in this particular experiment. And that white flower color he decided to call the recessive trait. So Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 different traits. And so over here on the right, if you notice in this chart, the top one here is going to be the flower color, which we've been looking at, the purple versus the white. He also looked at flower position. He looked at seed color, yellow versus green, seed shape, round versus wrinkled, pod shape, pod color, and then he also looked at something called stem length, in other words, tall versus dwarf. What Mendel called a heritable factor actually is what we call now a gene, and this is a word that you're probably familiar with again from Honors Bio. So Mendel actually developed a hypothesis to help explain the 3 to 1 inheritance pattern that he had observed after counting all of the F2 offspring in many of his experiments. 4 related concepts are going to make up this model or this hypothesis that he had created. And these concepts can be related to what we now know about genes and chromosomes. And we're going to discuss genes and chromosomes a little bit later in the year. So the first concept that he had looked at is that alternative versions of genes are going to account for the variations that we see in all inherited characters. So for example, when we looked at the color of the flowers in the pea plants, it actually existed in 2 versions. One, of course, was for purple and the other was for white. So these alternative versions of the gene are now what we call alleles. So again, alleles are going to be different varieties of that gene. So each gene is going to reside at a specific locus or a location on a specific chromosome. So if you look down here at the example, this band that you see, and this is considered a pair of homologous chromosomes. So when you talk about homologous, think back to our discussion of meiosis. When we talked about meiosis, of course, when you go through that particular process, you refer to a homologous pair. And a homologous pair basically means we have one chromosome that came from the mom, and we have one chromosome that came from the dad. But when you look at these chromosomes, they're exactly the same almost. In other words, these bands both actually code or determine flower color. But again, they could be different alleles because remember, there can be different varieties of that same gene. So in this case, on the mom chromosome, we have a band that's going to code for purple flowers. And of course, in this particular case, for this second chromosome, we have, and this one came from the dad, we have this band actually coding for white flowers on this chromosome. So remember, we have to kind of take some of the terminology that we had talked about or looked at in mitosis and meiosis and bring it into our discussion of genetics. So the second concept that he had determined was that for each character, an organism inherits, again, there are going to be two alleles. And again, as we had said, there's going to be one from each parent, and we had demonstrated that in our previous screen. Mendel made this deduction without really knowing about the role of chromosomes. So during his experiments, he really didn't have the knowledge of chromosomes to help him out. But based on what he had seen in his numbers and based on what he had seen in the outcome of his experiments, he was able to deduce that this was probably the case. The two alleles at a locus on a chromosome may be identical, as in the true breeding plants of Mendel's P generation. So in other words, maybe both chromosomes, again a homologous pair, one from the mom, one from the dad, maybe they both coded for purple flowers. Alternatively, the two alleles at a locus could possibly differ. And again, if you look at the F1 hybrids, remember that when you look at the offspring of those F1s, we got some purple flowers and we actually got some white flowers as well. So the third concept is that if the two alleles at a locus do differ, then one, the dominant allele, is going to determine the organism's appearance. In other words, it's going to overshadow the other allele. And the other is considered the recessive allele. And again, that's going to have no noticeable effect on the appearance because that dominant allele is again sort of overtaking the characteristic that's being coded for that recessive allele. So in the flower color example, the F1 plants had purple flowers because the allele for that trait is dominant. In other words, purple is dominant over white flower color. So the fourth concept, now known as the law of segregation, basically states that the two alleles for a heritable character is going to separate or segregate, in other words, separate from each other, during gamete formation. When we talk about gamete formation, we're talking about the formation of the sperm and the egg. And again, it's going to end up in different gametes because each gamete can only have one copy of that allele. But remember, we have two. Remember, we have a homologous pair. One came from our mom, one came from our dad. Thus, an egg or a sperm gets only one of the two alleles that are present in the somatic cells of an organism. So this segregation of alleles is going to correspond to the distribution of homologous chromosomes to different gametes, as we had said, that occurs during meiosis. Mendel's segregation model is going to account for the 3-to-1 ratio that he was going to observe and that we would observe if we carried out these same experiments. So in other words, if we let that F2 generation self-pollinate, or you could even take two of the individuals from the F2 generation and actually cross them, you're probably going to end up, unless there's something strange happening, with a 3-to-1 ratio. The possible combinations of sperm and egg is something that we would show using something called a Punnett square. And I know most of you have had some experience with Punnett squares. A diagram for predicting the results of a genetic cross is what this Punnett square is going to do for you. In other words, it's going to basically demonstrate the cross between individuals where we have a pretty solid idea of their genetic makeup. Now, a capital letter is going to represent the dominant allele in our Punnett square, and a lowercase letter is going to represent our recessive allele in that Punnett square. So this particular diagram is going to sort of illustrate what we've been talking about. So again, if we start with that P generation that you see right up here, this is going to be the genetic makeup, and that would be represented by these letters right here, of our two parental individuals. So in this case, when we're talking about the P generation, remember they're considered true breeding. This particular individual has purple flowers, and so it has two capital P's, and it has two because it has an allele from its mom and an allele from its dad. And this particular individual, the white flower, has two lowercase p's, and that's because this is considered the recessive characteristic when compared to the purple flowers, this dominant characteristic. And so when each of these flowers produces gametes, which remember are going to be the sperm or the egg in that individual, each sperm or egg is only going to have one copy of that allele. So in this case, the only copy they can pass down in purple flowers is the capital P. In the white flowers, the only copy it can pass down is going to be that lowercase p. Now, when those two gametes come together and produce offspring, that's going to represent the F1 generation. But if you notice, remember we had said purple flowers are dominant over white. So the only color variety you're going to get in that F1 generation is going to be purple flowers. But if you notice, when you look at their genetic makeup, you'll notice we have one capital P and one lowercase p. But again, that recessive allele characteristic does not show up. The white color does not show up because purple overtakes the white. Now, if you notice the gametes that are produced from these purple flowers half of them will produce a capital P and half the gametes will be a lowercase p. So there's a 50-50 chance of getting either allele. So when these F1 generations either self-pollinate, or what we could even do, we could cross-pollinate them, you're going to notice in this F2 generation, if we make a Punnett square to sort of illustrate what we're doing, what we would do is we would put the gametes on top and we would put the gametes on the left-hand side. So the sperm, again, capital P, lowercase p. So again, from one parent, we're going to have both possibilities. In other words, there's a 50-50 chance of either allele. And for the eggs, it's exactly the same because, again, this is the F1 generation and they have one of each. So there's a 50-50 chance of an egg either having a capital P or a lowercase p. And so then when you actually fill in your Punnett square, we simply take these two letters, put them together, and we do that for each one of the boxes. And then we basically just count is what we do. And so if you notice in this case, we have 3 purple to 1 white. So remember, when it comes down to genetics, if that capital P is there in this case, then we are going to get that dominant color. But if it's absent, then that's going to allow that recessive color to appear. And so this gives us the typical 3-to-1 ratio that we would find in the F1 generation cross. So some of the vocabulary that's going to be really useful to you as you go through our discussion of genetics is some of the words that you see on this screen in bold print. So when we talk about an organism that has 2 identical alleles for a character or a trait, it is said to be homozygous. Remember, this prefix HOMO is going to represent same. So we have 2 identical alleles. If it has 2 different alleles for a gene, it's said to be heterozygous for that trait. In other words, it has one of each. So in the previous screen, if you looked at the F1 generation, it had a capital P and a lowercase p. So again, that would have been considered heterozygous. Unlike homozygotes, heterozygotes are not considered true breeding. And they're not considered true breeding because if you think back to our previous example, when you take those F1 and you cross them, we do have an opportunity there to actually get that recessive trait. So Mendel's segregation model is going to account for the 3-to-1 ratio that he observed in the F2 generation of his numerous crosses that he had done with the different characters that he looked at in the garden peas. So the possible combinations of sperm and egg, again, can be shown by using the Punnett square like we had looked at before. And again, this Punnett square is simply a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. And so remember, the capital letter is going to represent the dominant and the lowercase is going to represent the recessive. And if you look at this screen, this is simply illustrating what we had discussed in the previous example. Again, talking about the homozygous condition. Again, looking at the prefix, it means the same. We have 2 of the same allele. The heterozygous condition, if you notice, we have 1 capital, 1 lowercase. You notice we have our ratios down here depending on what the cross was. And a couple of other terms that we did not introduce in the previous screen are up here at the top of this screen. So I want to make sure we talk about these real quick. One of those is phenotype and one of those is genotype. Again, most of you are probably familiar with these 2 terms, but just to review, again, a phenotype is going to be the physical representation of that trait. So in this case, with the flower color, that would be considered the phenotype, whether it's going to be purple or white. For genotype, we're looking at the genetic makeup of those alleles. So again, whether it's a capital P or a lowercase p when we look at the genetic makeup for that individual. Now, sometimes it's going to be unclear as to whether or not it's actually 2 capital P's or a capital P or lowercase p for an individual. In other words, if we had a purple flower, it'll be purple, but we won't know whether it's homozygous or heterozygous for that particular characteristic. So what we would do is we would do something called a test cross to determine that. So such an individual must have 1 dominant allele if it has that dominant characteristic. But again, the individual could be either homozygous or heterozygous. So what we would do is we would actually take that individual and cross it with a recessive phenotype. So again, remember the recessive can only have 2 lowercase p's. So the prediction of our Punnett squares would either be this or this. And so in other words, we would take the 2 capital P's, again cross it with the recessive, so 2 lowercase p's. Our expectation would be that all of the offspring would actually be purple flowers because again, if you look, they're all heterozygous. There's no possibility for getting the recessive color, in this case the white colored flower. But if you do the cross, and if you notice, again, we have the heterozygous here, and we have the recessive individual here. If we did the cross, then we would expect the offspring to be 50% purple and 50% white color in terms of the flower color for this particular example. Now, sometimes, again, this is all a matter of chance, so you never know quite what you're going to end up with when you actually do the cross. But if you did it enough times, then you would have this expectation if you take a heterozygous individual and cross it with a recessive. So if any of the offspring display the recessive phenotype, it has to be that heterozygous condition for that particular dominant phenotype that we're looking at. Now, another thing that Mendel had looked at, or actually derived from his many experiments with garden peas, was something called the law of independent assortment. And so Mendel derived, again, the law of segregation by following a single character, but this whole idea of the law of independent assortment actually involved two characters. So in this case, when you're just looking at segregation, the F1 offspring that was produced from his crosses were considered monohybrids, so that F1 offspring from that parental generation were considered monohybrids. Again, individuals that are heterozygous for one character. Again, when you cross those heterozygotes, that is considered a monohybrid cross. Again, Mendel identified his second law of inheritance by following two characters actually at the same time. So crossing two true breeding parents differing in two characters produces something called a dihybrid in that F1 generation. So there's two characteristics that we're looking at. And we would consider that F1 heterozygous for both characters. A dihybrid cross, a cross again between two F1 dihybrids, can determine whether or not two characters are transmitted to the offspring as a package, in other words, together, or actually transmitted independently of each other. So in Mendel's experiment, as you can see on this screen, this is what he did. What he was doing is he was looking at the color of the pea, so in this case yellow versus green, and then he was also looking at the texture of the peas. So he was looking at the smooth coat of the pea versus the wrinkled coat of a pea. And if you notice the pea generation, again, they're both considered true breeding for each characteristic, and the gametes that are produced, again, you have to follow both traits through, and so you're looking at two different characteristics in each of the gametes. So the Y is going to represent the color, and the texture is going to be represented by the R. So as we make our way down, when you have the F1 generation, remember they're going to be considered dihybrids. Di means two, so we have two different characteristics, and they're hybrids, so they actually have the capital letter and the lowercase letter for each of the different traits we're looking at. But when you look down here, you're going to notice that in terms of independent assortment, what we have to think about here is we have to think about what are we going to end up with if we actually cross two of these together. So when you look at the F2 generation in this case, you're going to notice that if it was not independent assortment, in other words, it was dependent assortment, in other words, they kind of influenced each other as they were passed on or actually combined to produce that offspring, you would end up with a typical phenotypic ratio of about 3:1. But if it's independent assortment, you're going to notice that we have a lot more different opportunities to get different combinations for those offspring. And based on the numbers that he had collected from his many experiments with dihybrid crosses, he found that phenotypically you ended up with a ratio for whatever characteristics you were looking at. Again, you were looking at two, a 9:3 to 3:1 ratio. And so based on his results, this basically proved to him that these characters, in other words, the color versus the texture, in this case, of the seeds, assorted independently during the cross. In other words, they had no influence on each other as they were sort of put back together to produce those offspring. So again, using a dihybrid cross, Mendel developed the law of independent assortment. So again, the law of independent assortment is going to state that each pair of alleles is going to segregate independently of each other during gamete formation. You know, strictly speaking, this law applies only to genes on different non-homologous chromosomes. And the reason for that is because genes that are located near each other on the same chromosome, again, tend to be inherited together. And that's something we're going to look at a little bit later in chapter 14. So during our discussion of genetics, again, we've been looking at a lot of numbers. And so what we have to think about here is that the law of probability has a big influence in Mendelian inheritance. So Mendel's law of segregation and independent assortment basically reflects the rules of probability that most of you probably learned in math. So again, when tossing a coin, and this is an example that's commonly used in a science class, the outcome of one toss really is going to have no impact on the outcome of the next cross. So what that means basically is if you toss a coin and it comes up heads, and then you toss it again and it comes up heads, and you toss it again and it comes up heads, that doesn't necessarily mean that, okay, well, it's came up heads 3 times. Of course, that fourth toss is going to come up heads. Each one of those events is an independent event, and it has no impact on the previous or the future events. So in the same way, the alleles of one gene is going to segregate into gametes independently of another gene's alleles. In other words, there's no influence on each other. The multiplication rule states that the probability that 2 or more independent events will occur together is simply the product of their individual probabilities. So if you notice on this screen, it says the rule of addition is going to state that the probability that any one of 2 or more exclusive events will occur is going to be calculated by adding together their individual probabilities. And so in this case, the chance of at least 2 recessive traits occurring in this particular example, and I can't tell you for sure what these letters are representing, but if you notice, the chance of at least 2 recessive traits being represented in each of these different genotypes is about 1/16 based on what we see on the left-hand side. And if you notice, for each of these different scenarios, if you just take all of those 1/16 and 2/16 that you see on the right and you simply add them together, the chance of at least 2 recessive traits when presented with this type of situation is actually 6 out of 16. So you have to take each one of these independent events and simply add these events together. All right, so that's going to finish up our very first screencast for Chapter 14. As always, please make sure that you have completed your screencast study guide.